Hello everyone and welcome to the biology lecture series, and today we'll be discussing the topic sexual reproduction in the flowering plant. Okay, so first of all I will give you a brief introduction about the flower, okay, and the parts of the flower, all right, and then we'll study the rest of the topics. Okay, so let's start with this lecture. So you must have seen the flowers. The flowers, they come in variety of the colors. If we talk about the petals of the flower, the petals of the flowers, they are of different colors, okay, and you enjoy gazing at them, all right. And the flower, they also produce particular type of scent, okay, and perfumes as well. And there are rich colors present in the petals that are basically, what is the function of that rich colors of the petals? It is there to attract the human being as well as to attract the pollinators also. Okay, so whenever you see the flower, you keep on gazing at it, you smell it also, okay, and you have a look at the color pattern of the flower. Okay, so all these the scent, the perfumes, as well as the colors of the petals, they all of these features, they aid in the sexual reproduction. Okay, because all of these, they attract the pollinators. All right, pollination, we will talk about it in later classes. As for now, you have to just keep in mind that these are the features that help to attract the pollinators, and then pollination is possible. What is pollination? Pollination is the transfer of the pollen grains from the anther to the stigma. Okay, we'll talk about anthers also and the carpels also. Now flowers do not exist only for to be used for the own selfishness needs, for human purpose only. Okay, now all flowering plants, they show sexual reproduction. Clear? So flowering plants, they belong to what? They belong to angiosperms. Flowering plant, they belong to angiosperms. Okay, next. Now a look at the diversity of structures of the inflorescence, flowers, and the floral petals shows an amazing range of adaptations to ensure formation of the end products of sexual reproduction, fruits and the seeds. Okay, now whenever you look at the flowers, you can see there are a lot of variations. Okay, there is lot of variation in the inflorescence. You must have studied there are different type of inflorescence, racemose inflorescence and cymose inflorescence. Okay, there is a diversity in the structure of the floral parts. Okay, what are floral parts? Floral parts are the petals, sepals, androecium and the gynoecium. Okay, now these floral parts, okay, we can write here what are the floral parts. So flowers typically has 4 whorls. There are 4 whorls present in the flower. Okay, and the outermost whorl is known as the sepal. Okay, and the collection of the sepals is known as the calyx. So this is the outermost. Then inner to sepals we have petals. Okay, then inner to petals androecium is present. Okay, we can write androecium represents the stamens. Stamens is the male reproductive part. Okay, what are petals? Petals are the collection of petals is known as corolla. Then stamen, androecium. Okay, androecium represents the male reproductive structure. Okay, and stamen represents the androecium. Then last one is the gynoecium. Gynoecium, it is the pistil. Okay, clear? So there are four whorls: sepals, petals, stamens, and the pistil. Okay, so there are a lot of variation in the floral parts also. Okay. Now these diversity in the structure of the flower is because of the adaptations. Okay, to ensure formation of the end product of the sexual reproduction. What is the end product of sexual reproduction? It is the fruit and the seeds. Okay, so we'll study later on that after the fertilization, what happens after fertilization? Ovary develops into fruits and ovule develops into seeds. All right, clear? We'll study about this later. How ovary develops into fruit, how ovules develop into seeds. So these are the end product of the sexual reproduction. Now in this chapter, let us understand the morphology, structure, and the process of the sexual reproduction in the flowering plants. Okay, we'll study about the morphology, structure, as well as the sexual reproduction in the flowering plants, which are known as the angiosperms. Now, human beings have had an intimate relation with the flowers since the time immemorial. Okay, human beings, and it means the humans, they have a very intimate relationship with the flowers. Okay, now flowers are the object of aesthetic, ornamental, social, religious, and the cultural value. So these flowers, they have a lot of value, they have a lot of aesthetic value, they have a lot of ornamental value, they have social value, they have religious value also. Okay, as well as the cultural value. Now they have always been used as symbols for conveying important human feelings such as love, affection, happiness, grief, mourning. All right, so that's why there is a very intimate relationship between the humans as well as the flowers. So you can see flowers are used. Okay, there are a lot of uses of these flowers. They have aesthetic, ornamental, social, religious, and cultural value. And these flowers are also used for conveying the human feelings such as love, affection, happiness, grief, and mourning. Clear? Now in this figure, you can see figure 1.1 shows you the parts of a typical flower. Clear? So I've already told you what are the four whorls of the flower. Outermost is the sepals, known as the calyx. Then inner to it is sepals. Okay, then we have the androecium, and in the center we have the gynoecium. Okay, so here you can see sepals. Okay, and the collection of the sepals is known as the calyx. Clear? Now, outermost is the sepals. Then we have the petals. Collection of petals is known as corolla. Then inner two petals we have the androecium, which is represented by the stamen. Okay, so stamen consists of anther and the filament. So this is a stamen. Clear? We can draw a stamen like this. Okay, so these two are known as the anthers, and this is the filament. And this entire thing is known as the stamen. Okay, stamen, or you can say androecium, okay, represents the male reproductive system, and androecium consists of the stamens. Clear? Then inner to this androecium we have the gynoecium. Okay, so you can see here, this is the ovary, this is the style, as well as the stigma. So this is gynoecium. We were talking about androecium. Now let's talk about the gynoecium. So gynoecium is like this. So there is a receptive part, okay, and then there is a tube, and then we have the swollen base, okay. So this is the swollen base, which is known as the ovary. Now this part, this is known as the style, which is a tube-like structure, and this is known as the stigma. Okay, this is known as the stigma. Stigma is the receptive part. It is the receptive part, means it receives the pollen. All right, clear? So this is the image showing you the parts of the flower. You can see ovary, which is the swollen base, and then this is the style, and this is the stigma. Now this entire ovary, style, and stigma, this is known as the pistil. Okay, this is the pistil, or you can say carpel. Okay, and the gynoecium, which is the female reproductive part, this is represented by this pistil or the carpel. Clear? Now let's move on. Let's study about the pre-fertilization events. Okay, means before fertilization. What are the structures and the events before the fertilization? Now, before fertilization, what happens? There is the production of the gametes. Okay, so events are like this. First is the pre-fertilization event. Then, after pre-fertilization, we have the fertilization events. And then, post-fertilization event. Clear? Pre-fertilization, fertilization, and then post-fertilization. So pre-fertilization, what happens? Formation of, you can say, gametes. Fertilization is the union of gametes. And then post-fertilization will be development of the embryo. Okay, so we'll talk about this in detail. First, let's study about the pre-fertilization. Now, much before the actual flower is seen on the plant, the decision that the plant is going to flower has been taken. Okay, so if you know about the phases of the growth, or you can say phases of the growth of the plant. So first is what? First is the vegetative phase. Okay, and second one is the reproductive phase. Okay, so there are two phases. First is the vegetative. Second is the reproductive. So during the reproductive phase, what is formed? Flower. Flowers are produced during the reproductive phase. Clear? Now several hormones and structural changes are initiated which leads to the differentiation and further development of the floral primordium. So for the formation of the flower, there is a need for the formation of a floral primordium. So a lot of structural changes takes place, as well as there are some hormones are also required in the formation of the floral primordia. Okay, some of the examples of the hormones are auxins, cytokinins, then we have gibberellins. So these are involved in the formation of the flower. The next is inflorescences are formed which bear the floral buds and then the flower. Okay, inflorescence what? It is the collection of the flowers on the axis, or you can say arrangement of the flowers on the axis. This is known as the inflorescence. What is the inflorescence? It is the arrangement of flowers on the main axis. Clear? The inflorescences are formed which bear the floral buds and then the flowers. Alright, next. Now in the flower, the male and the female reproductive structures, the androecium and the gynoecium, differentiate and develop. Okay, so in the flower, the male and female reproductive structures. So male reproductive structure is known as the androecium and female reproductive structure is known as the gynoecium. And I've already told you, stamens represent the androecium and pistil or carpus represent the gynoecium. Alright, now we can recollect that androecium consists of a whorl of the stamens representing the male reproductive organ, and the gynoecium represents the female reproductive organ. Okay, next, now this is a diagram, okay, which shows the two parts of the typical stamen. So stamen, so you can see this image A. Stamen consists of a filament and a anther. Alright, and then the terminal generally bilobed structure called the anther. Okay, so this structure is known as the anther and it is generally bilobed. Okay, you can see here it is bilobed, two lobes are present and each lobe contains two pollen sac. Okay, clear? So this was the filament or the stalk of the, you can say, the stamen. Now each lobe, it has two lobes. Okay, so anther is bilobed, means it has two lobes and each lobe, it contains two pollen sac. Okay, clear? Now this pollen sac consists of pollens. Now the proximal end of the filament is attached to the thalamus of the or the petal of the flower. Okay, so what is thalamus? You should know thalamus is the swollen part. Okay, it is the swollen basal part to which the floral parts are attached. Okay, so the proximal end of the filament, you know, stamen consists of anther as well as the filament. So the filament is attached to the thalamus or the petal of the flower. Okay, thalamus is the swollen part, okay, of the, you can say, pedicel. Clear? Now next, what is next question? The number and length of the stamens are variable in flowers of different species. Now if you were to collect a stamen each from 10 flowers, okay, and these flowers belong to different species, and arrange them on a slide, you would be able to appreciate large variation. So if you collect these stamens from flowers that belong to different species, then you can see there is a lot of there is a lot of variation in the type of the anthers. Okay, clear? Now this is an image again showing you the anther and the two lobes of the anther as well as the pollen sac. Now typical angiosperm anther is bilobed with each lobe having two theca. So each lobe consists of two theca. That's why they are known as ditheca. So di means two. So if there is a presence of two theca, it is known as dithecus. So each lobe consists of two theca. Okay, or you can say each lobe consists of two pollen grains, two pollen sacs. Now in a typical angiosperm anther, which is the male reproductive organ, okay, anthers are the male reproductive organ, pollen grains are produced in it, and there are typically two lobes, okay, which are joined together by a connective tissue. Now each lobe usually contains two pollen sac. Okay, each lobe contains two pollen sacs, also known as microsporangia or the theca. Okay, so these pollen sacs are known as microsporangia or theca. So you have to remember these terms. This is very important. Now the pollen sacs are the site of pollen grain production through the process of the meiosis. Okay, so pollen grains are produced through meiosis. The pollen grain should be haploid. So in total, a bilobed anther typically contains 4 pollen sacs with 2 sacs in each lobe. Clear? So there are 2 lobes, and within each lobe we have 2 pollen sacs, and within the pollen sacs the pollen grains are present. Clear? Next, now here is the image. Okay, so now let us understand the various type of tissues and their organization in the transverse section of the anther. So this image shows you the transverse section of the anther. Okay, so you can see here the layers present in the anther. Epidermis is present, which is the outermost layer. The inner to epidermis we have the endothecium. Inner to endothecium, middle layers are present. Okay, and then the innermost layer is known as the tapetum. Okay, and you can see the anther is bilobed here, and each lobe consists of 2 pollen sacs. And here you can see the connective tissue also. Here again you can see the layers. All right, clear? And here also you can see the dehiscence of anther is showing here, and release of pollen grains is also observed. The pollen grains are being released here. Pollen grains are haploid structure as they are produced by the process of meiosis. Okay, so this is clear. Now the bilobed nature of the anther is very distinct in the transverse section of the anther. Okay, whenever you cut the transverse section of the anther, you can see the bilobed anther. Now anther is a 4-sided structure. It is a tetragonal structure consisting of 4 microsporangia. What were microsporangia? Microsporangia were the pollen sacs located at the corners, 2 in each lobe. Clear? Next is the microsporangia develop further and become the pollen sacs. So microsporangia will develop into pollen sacs. Clear? Now they extend longitudinally all through the length of the anther and are packed with the pollen grains. The pollen sacs consist of the pollen grains. Now structure of the microsporangium. In a transverse section, a typical microsporangium appears near circular in outline. So microsporangium which develops into pollen sacs, this appears circular. Okay, clear? Nearly circular. Again, this is an image I've already shown you. Here you can see 4 walls of the anther: epidermis, endothecium, middle layer, and the tapetum. So epidermis is the outermost layer and the tapetum is the innermost layer. Epidermis will be the outermost and the tapetum is the innermost. Okay, clear? Now next, the outer 3 wall layers perform the function of protection and help in dehiscence of anther to release the pollen. Clear? Outer 3 walls. Okay, outer 3 walls layer perform the function of protection. What were the outer 3 walls? Epidermis, endothecium, and the middle layers. So they provide protection and help in the dehiscence of the anther as well. Dehiscence of anther means release of the pollen. Now the innermost layer, which is known as the tapetum, what is the function of the tapetum? It helps in the nourishment of the pollen grains. Now the cells of the tapetum possesses dense cytoplasm and generally have more than 1 nucleus. Okay, now there is a question: can you think how a tapetal cell would become binucleate? Now when the anther is young, a group of compactly arranged homogeneous cells known as the sporogenous tissue, it occupies the center of the each microsporangium. Clear? Now microsporogenesis, let's study about the microsporogenesis. Okay, now as the anther develops, the cells of the sporogenous tissue undergo meiotic division. So sporogenous cells should be diploid because meiosis usually takes place in the diploid cells. Okay, so there is meiosis in the sporogenous tissue, okay, which leads to the formation of microspore tetrads. Tetrad means 4 cells. 4 cells are formed in the tetrad. Clear? So we are talking about microsporogenesis here. Now next. Now what will be the ploidy of the cells in the tetrad? They will be haploid. Okay, because they are produced by meiosis. So they will be haploid, means they contain one set of chromosome. Now, as each cell of the sporogenous tissue is capable of giving rise to a microspore tetrad, clear? So each cell of the sporogenous tissue which is diploid, it can undergo meiosis and it can give rise to a microspore tetrad. Now, each one is potential pollen or microspore mother cell. Clear? Now, the process of formation of microspore from the pollen mother cell through meiosis, this is known as microgenesis. So, how can we define microgenesis? It is the formation of the microspores from the pollen mother cells, also known as the microspore mother cells. Clear? This is known as the microsporogenesis. Now, the microspores, as they are formed, are arranged in a cluster of 4, which is known as the microspore tetrad. Now, as the anthers mature and dehydrate, the microspore dissociate from each other and develop into the pollen grains. So, microspores, in the end, they are transformed into the pollen grains. Okay, clear? Now, here in this image, you can see inside each microsporangium. Okay, what was microsporangium? Microsporangium later developed into the pollen sacs. Thousands of microspore or pollen grains are formed that are released within the dehiscence of the anther. Of this image C, you can see after the dehiscence of the anther, pollen grains are being released. Now next, let's talk about the pollen grain. What is a pollen grain? The pollen grain represents the male gametophyte. So if someone asks you what is a male gametophyte, so male gametophyte, okay, it is pollen grains. Pollen grains represents the male gametophyte. Clear? Now, if you touch the open anthers of the hibiscus or any other flower, you would find deposition of yellowish powdery pollen grains on your fingers. So that powder consists of the pollen grains. Okay. Now, sprinkle these grains on a drop of water, taken on a glass slide and observed it under the microscope. So what you have to do? There is one exercise. You have to sprinkle these grains on top of the water. Okay, sprinkle some of these pollen grains on top of the water, which is taken on a glass slide, and then you have to observe it under the microscope, and then you can tell about the structure of the pollen grains. Okay, so here is the images. So you will be amazed, okay, at the variety of the architecture. Means, so there is a lot of variation in the size, shape, color, as well as the design of the pollen grains from different species. Clear? Now here you can see, this is the scanning electron microscope, SEM. Scanning electron microscope tell you about the surface. Okay, so these are the pollen grains. These images, you can see the structure of the pollen grain is so different. Alright, so there is a lot of diversity in the size, shape, color. You can see there is a difference in the color also. Okay, so this much variation is seen in the pollen grains. Now, pollen grains are generally spherical, measuring about 25 to 50 micrometers in the diameter. It has a prominent two-layered wall. It has 2 layers, or it is surrounded by 2 layers. The harder outer layer is known as the exine. Okay, this exine is not easily degradable by the enzymes, which is made up of the sporulin. So that's why it is made up of the sporulin, which is very hard material. So it is not degraded by the enzymes. Okay, which is one of the most resistant organic material known. Now it can withstand high temperature as well as strong acid and the alkali. And no enzyme is known that can degrade sporopollenin so far. So you can see sporopollenin is, okay, it is a very, you can say, hard substance, and it is not able to degrade easily. Okay, clear? Now let's move on. Now, pollen grain exine has prominent aperture called the germ pore, where sporopollenin is absent. So you know that the outer layer is known as exine, internal layer is known as intine. The pollen grain's exine has prominent aperture. It has a prominent aperture called the germ pores. These are known as the germ pores, where sporopollenin is absent. Now, pollen grains are well preserved as fossils because of the presence of the sporopollenins. Sporopollenins is very resistant material. It cannot be degraded by acid, alkali or the enzymes. That's why pollen grains are well preserved as the fossils. The exine exhibit a fascinated array of patterns as well as the design. Okay. Now, why do you think the exine should be hard? Why it should be hard? And what is the function of the germ pore? The inner wall of the pollen grain, is known as the intine. So, outer wall is known as the exine, inner wall is known as the intine, and you have to find out the answer. Okay, why the exine is so hard? Answer is exine first of all is made up of sporodin, and why it should be hard? Okay, because the pollens, they have to travel a lot of distance and they are not able to reach their target. They are not able to reach the stigma of the another flower. Say if you talk about the cross pollination, these pollen grains have to travel a lot, okay, and they can be degraded while before getting transferred to the stigma of the another flower. Okay, that's why they are covered with sporopollen, because they have to face very harsh conditions. Clear? Now next, in a thin and continuous layer, what is intine? Intine is a thin and continuous layer made up of cellulose as well as the pectin. The cytoplasm pollen grain is surrounded by a plasma membrane. This also you have to remember. So we're talking about the structure of the pollen grain. So what is the outer layer? Exine, which is very resistant because of the presence of sporopollen. Inner layer is known as intine, and the pollen cytoplasm, it is surrounded by a plasma membrane. Now this is an image showing you the pollen grain, and the pollen grain are usually released at the 2-cell stage. They are usually released at the 2-cell stage. Okay, the larger cell is known as the vegetative cell. The smaller cell, this is known as the generative cell. Okay, in some cases, the pollens are released in the 3-cell stage. Okay, where the generative cell divides to form the 2 pollen grains. Okay, now the vegetative cell is bigger, as I have told you, and has abundant food reserve and are largely irregular and have large irregularly shaped nucleus. The generative cell is small and floats in the cytoplasm of the vegetative cell. Now it is spindle shaped and dense cytoplasm is present as well as a nucleus is present. Now in above 60% of the angiosperm, pollen grains are shed at the 2-cell stage. So this also you have to keep in mind. Over 60% of the angiosperms, pollen grains are released in 3-cell state. Sometimes they are also released at the 3-cell stages. Okay, so what happens? One is the vegetative cell, which is large, and here the generative cell divides into 2 gametes. Clear? Clear? So this is known as the 3-cell stage. Okay, one is the larger vegetative cell, and the other generative cell divides to form the 2 male gametes. Now in the remaining species, generative cell divide mitotically to give rise to 2 male gametes. So this is known as the 3-cell stage. Now the pollen grain of many species causes allergies also. So, okay, one of them is known as the hay fever also. Asthma is also caused. So pollen grains of many species cause several severe allergies and bronchial afflictions. Okay, so these are this leads to the chronic respiratory disorders. Some of the examples are asthma as well as the bronchitis. Now it may be mentioned that Parthenium, also known as the carrot glass, now that come into India as a contaminant with imported wheat. Clear? Now has become ubiquitous in occurrence and cause pollen allergy. So Parthenium you must have seen. Parthenium you must have seen on the roadside. It is a weed actually. Parthenium is a weed now. It is an unwanted plant. So in the early days, this Parthenium, it came along with the imported wheat. So we were importing wheat from the abroad, and along with this, we got this Parthenium weed. Okay, and now the pollens of this Parthenium, they causes lot of allergic reactions. And one more thing about pollen grains are rich in the nutrients. Now it has become a fashion in recent years to use pollen tablets as the food supplements. So pollen tablets are also used as the supplements because it contains lot of nutrition. Now in Western countries, a large number of pollen products in the form of tablets, syrups, these are available in the market. So this is an example of the pollen products. Here, this is the bee pollen. So the pollen consumption has been claimed to increase the performance of the athletes as well as the race horses. Both athletes and horses, they require a lot of nutrition, and this nutrition is available in this pollens. And these are the products you can see. Bee pollen, and these are some of the more other pollen products. So these are obtained from the pollens. Now, once they are shed, pollen grains have to land on the stigma. So this is known as the pollination. The transfer of the pollens from the anther to the stigma of the flower, and they have to be transferred. Pollen grains has to be transferred to the stigma before they lose their viability. Clear? Now, how the pollen grains, they maintain their viability? So the period for which pollen grains remain viable is highly variable and to some extent depend on the temperature as well as the humidity. Now in some cereals such as the rice as well as the wheat, pollen grain lose viability within 30 minutes of their release. Okay, and in some members of the Rosaceae, Leguminaceae and Solanaceae, they maintain viability for months. Now you may have heard of storing semen sperms of many animals including humans for artificial insemination. Okay, so sperms and semen are also stored for artificial insemination. Now similarly, the pollens can also be stored. So pollens of large number of species can be stored for so many years in liquid nitrogen. What is the temperature of liquid nitrogen? 196 degrees Celsius. This is the temperature of the liquid nitrogen, minus 196 degrees Celsius. The pollen grains can be stored at this temperature for many years. Now such pollens can be used as pollen banks similar to the seed banks in crop breeding programs. Okay, so we'll end this session here. I hope you enjoyed this session and we will continue in the next lecture. Thank you very much for listening.